All right, everybody. Thanks so much for coming out tonight. Go ahead and get started. So first off, just like to thank all of our sponsors. So especially our platinum sponsor, Raven, and they're going to come up and give us a short presentation to kick things off tonight. We'll also just say the sponsors are the reason why we have all these nice food and drinks available. So huge debt of gratitude to the folks that make all this possible.
Oh, cool. Well, huge debt of gratitude to Chris, Sergey, and Nick, who actually run this seminar series and keep the community alive in Boston. So I am Jacob Oppenheim. I'm a venture partner at Raven. We are the venture incubator arm of RA Capital. RA Capital is one of the largest funders in biopharma. We fund everything from small newcos all the way up to public stage biopharma companies. We actually don't normally talk about ourselves all as much as we should, but we have been doing this since 2019. We have built 35 separate companies, 3 asset spinouts from pharma, 5 IPOs, 2 acquisitions, and we've been relatively successful at this. Some of our portfolios down here, some of these names might be familiar, some of these names might not be familiar. The reason I'm talking to you about all of this today is that we are continually building new companies, and we are very much invested in this idea that we can engineer biology today. I remember when I was in college, even when I was in graduate school, a lot of drug development was, well, do I have a probe here? Can I hit this target? Is there any way I could drug this or possibly even measure this thing? And today, really, I can come up with 5 or 6 different modalities if I wanted to drug a protein in the body, right? And all of us in this room here know about all the really cool and different ways that we can computationally and experimentally design ways of binding to, modifying the effect of, or otherwise neutralizing or upping the expression of a protein of interest. Because of this, we're starting lots of new companies in this space. So part of what we do is I run a small computational team within Raven supporting the creation of lots of newcos. Cassie is on my team leading protein design. We're hiring there. We are also starting these companies, a lot of which are in protein design or have strong protein design elements. And we're going to want to hire teams of great people into these companies when we start them. I just want to highlight really, really quickly, one of these portfolio companies founded by Rob Nickel, who built out the high throughput sequencing team at the Broad. He's a venture partner with us. Skyhook Bio is a integrated design, build, test, learn company for generating novel peptides and peptide therapeutics. There's an integrated small computational team. There is massive scale programmable synthesis at 10 to the 9th scale for testing and synthesis, which is pretty amazing, and 1-week cycle times. So we're using this to fund the creation of lots of different newcos and lots of different therapeutics companies. And lastly, we're hiring. Cassie?
Yes, we are hiring. So there are several different companies already within the portfolio that need protein design support, as well as very rapidly evolving ideas that we currently don't have the staff to keep up with. So we need to grow the team to work on these cool ideas. And just to give you kind of a flavor, in addition to having access to a really powerful ultra high throughput screening platform, right? We all know our designs are only as good as the results validating them. We also get to work on a wide variety of design approaches and protein problems. So this ranges from really targeted redesign, trying to balance multiple requirements such as immunogenicity, solubility, serum half-life, things like that, as well as really using protein designs more as a tool and being able to use our full toolkit, including fully de novo design. So it's a really fun place to work. It's very fast-paced. You get a wide range of exposure and really early stage development of a variety of companies. So we'll be here the whole seminar. Come up and talk to me afterwards if you have questions or you're interested. And we also have a QR code for the specific position we have posted right now. Thanks.
Thanks, everyone.
All right, thanks so much. We'll just also bump, for those of you who don't know, Cassie, she's a David Baker alumni and also been a part of this community for quite a few years now, and also one of our speakers too. Gave a talk, I think, back in the pre-pandemic days before everything was recorded and online. So unfortunately, you can't go watch her talk, but still highly recommend. Okay, very excited to have Seth Fraden with us today to give the seminar. So Seth is faculty at Brandeis and has been in the DNA origami community for a very long time and has been folding macromolecules and has recently become excited about folding proteins and designing proteins as well. So looking forward to your talk. Please take it away.
Well, thank you very much to Nick, Chris, and Sergey for giving me this opportunity to address you. I am new to the protein community, and it's been a wonderful opportunity for me to be in this lecture and be able to ask many annoying and naive questions. So thank you for your toleration of that. And in reciprocal, this is a topic that's maybe outside of your familiarity, so please reciprocate with questions. I won't find them annoying, I promise. The team, I started on this project about 7 years ago with a sabbatical to Germany, to the lab of Hendrik Dietz, where he taught me this origami technology, and then I exported it from Germany to Waltham, where Brandeis is located. And then we've built a large team around our Materials Research Science and Engineering Center, our MERSEC, and the work that I'll present here comes from several groups. And of course, the postdocs, some of whom are here, Wei Xiao, who worked extensively on it. The outline of the talk is I'll first introduce self-assembly and focus on a test case of virus structure. So starting with proteins as the motivation for this work in DNA origami. And then I want to highlight the theory of the Caspar-Klug quasi-equivalence, the theory of structural biology of viruses. And that underlies all of our work. And then I'll go into the DNA origami technology and an example for antiviral therapy that's being commercialized. And then turn to a second kind of viral structure, the cylindrical filamentous viruses, and then their origami implementation, and then conclude with some lessons learned from the work in origami that our hypothesis is that they're transferable to protein assemblies. And so I would be curious to see if you think that's just a hallucination like you often get in AI, or if there's any merit to that concept. So, the PDB is full of single protein structures, but to be functional, proteins have to assemble into large units. And the question is, can we de novo make large protein assemblies, with high yield and precision? And so kind of two routes that nature takes in assembly. One is called addressable, where you have, every piece is different and there's lock and key driving the assembly. And the ribosome is an example, some huge machine that does unbelievable things. And it's, I don't know how many, 14 different proteins. Each one is unique and then assembles together. We're going to take the opposite kind of view, where we're going to take, like, say, a single building block that is designed, that has instructions for assembly encoded into that one block to make a large assembly. And this is what the viruses do. And so that's a very economical way to make a large structure, while the ribosome, you can make a very precise, highly functional, extraordinary machine. The cost is that each of the elements has to be unique and bespoke. So all subunits are unique in the addressable, and in the other extreme, you take a minimum number of subunits and you use many copies of them in order to create your structure. And so what are the attributes that these building blocks have to have? So one is valence. It's how many bonds per block. So one of these puzzle pieces is an individual block. And so that one has 2 bonds, one here and one here. Another element, attribute, are the specificity. How many of the bonds are distinct? So here you have a key and a lock. There's a recession and a protrusion, and one fits into the other. It's a puzzle piece, lock and key. So on this piece, there's 2 distinct bonds. The lock can't bind to another lock. It's only lock and key. We want to be able to make our building blocks in an arbitrary shape. And we want to control the binding angle, which means we need to also control the mechanical properties, the rigidity. It's one thing to make a bond with an average angle of 43 degrees, but if it's highly flexible, then you're going to run into design challenges. So this was the construction that we made out of DNA origami, where each cylinder represents a double helix of DNA. And so this triangle has 3 sides, very prosaically labeled side 1, side 2, and side 3. And how many bonds? Bonds are pairway, so 3 choose 2 is 6. There's 6 possible bonds. And in this example, we engineer the bonding so that only side 1, the red, binds to the blue, and side 3 binds to itself. Side 3 binds to side 3, and so we design it so that with interaction specificity. In this case, we designed it 2, but we could design so all 6 were active, or only 1 was active, or 3 were active. These are engineers have the capability of picking which interactions you want to encode. And so then historically, this kind of systematic study of icosahedral domes goes back to the 50s with Buckminster Fuller. Buckminster Fuller was an architect and a futurist, a futurist back in the 1950s. And he popularized these geodesic domes. This is the largest one that he built, 200 meters in diameter, in Montreal. And here's Buckminster Fuller. Buckminster Fuller went to school at Harvard. He was an undergraduate. He's known perhaps best for being expelled twice. The first time was when he brought a vaudeville troupe into his dorm room, and it was apparently very raucous, and he was immediately expelled, or the next morning was expelled. And the second time, after he was readmitted, he was expelled for lackadaisical attitude. And so in the 50s, during the Cold War, there was a World Fair in Kabul, Afghanistan, in the US at the last minute. I don't know, I guess some things never change. First they said they weren't going to go to Kabul, then they said they were going to go. So at the last minute, they decided to go, and they needed somebody to assemble a 100-meter diameter building that it had to be all in parts pre-manufactured in the United States, shipped on a military plane to Afghanistan, and then assembled with unskilled laborers. So Buckminster Fuller got the contract, and the idea was you have a hub. Here's these hubs, and you have holes in the hubs and poles, and you just stick the pole in the hub. And then, when the workers started working on it, they thought they'd get some square rectangular building, and instead they got this yurt. And the reason they said that it could be built without any errors was that, of course, a yurt is their cultural heritage, and it was natural for them to make such an object. They didn't need any instructions. And at the same time, Donald Caspar was working at Children's Hospital, not 100 meters from here. Here is a picture of him in his office, and he was working on the structure of viruses. And they had some X-ray data, diffraction data, and EM, the first EM pictures that indicated they were icosahedral. And he had seen Buckminster Fuller's domes built around. And here he is in Buckminster Fuller lived in Boston, and here's Caspar. He went over to his home and showed him his visualizations, his imaginations of what the viruses would look like, and worked out a theory with Aaron Klug, which Klug got the Nobel Prize for the virus structure. So Caspar was exploring the analogy across these length scales of 7 orders of magnitude between meter scale for architecture and nanometer scale for viruses. And the point that Caspar was focusing on is that in both cases, the domes were assembled without any architectural plans. There was just local rules. You've got this hub and you've got these poles, and the hub has holes in it. And some of the hubs have 6 holes, some have 5, and you shove the pole into these hubs. And then the structure and this is how viruses work. There's no architect telling the hundreds of proteins in the shell how to assemble. They come together willy-nilly, and a virus comes out. And so Caspar worked out this theory, and every virus found until the 80s fell into this category of theory, which I'll go into in a few minutes. And the challenge that the viruses needed to overcome is that they have to enclose the genome that encodes the capsule. And so if you're this virus, there's 200 proteins. If you have different proteins for each of those 200, it's 200 genes. They're never going to fit in that tiny space. So that's the evolutionary challenge that they had to, and their solution was make it out of the minimum number of components. And it's Caspar who solved the mathematical theory for what is that minimum number of components. I'll just briefly walk you through that. So one of the structures is that the architecture that all these viruses adopt is a icosahedral. And so an icosahedron, one of the 5 platonic solids, has 20 faces, and you can map a protein onto each face of the triangle, where at each vertex you have a protein. So each triangle corresponds to 3 proteins. 3 times 20 is 60. So 60 proteins, like this K9 K9 something virus, CPV, just K9 something virus, 60 identical copies. But many capsids have multiple of 60. You never find a virus with 61. You never find it with 62. It's multiples of 60. So CCMV, chlorotic mosaic virus, it has 180 subunits. So what's going on? Why is there this quantization and this numerology? And so Caspar and Klug in 62 worked out the theory for it. So they say, imagine a 2-dimensional lattice, and they said, it's a triangular lattice, and each protein you represent as a sphere. And if they're all identical, they're identical spheres. So you have this closed pack of spheres. And then define a lattice K and H on this hexagonal lattice triangular net. And then imagine you cut out one triangle. One triangle has 3 proteins in it. And now you cut out 20 triangles. Here's one triangle, cut out 20. And so this isn't origami, this is Chirality. You cut out the 20 and then you fold it up so all the edges meet themselves. So these 5 edges come together to form a pentamer. These 5 edges come together, and then this line you take some Scotch tape and seal with this line. Then you have an enclosed object, and this is what the enclosed object looks like. It's a icosahedra, 20 identical triangles. All sides are equivalent. Every point in the triangle is equivalent. And so then how does this icosahedral virus, satellite tobacco necrosis virus, how does this monomer, shown on the left, know to assemble into an icosahedron with exactly 60 copies of itself? And so this is a drawing from Caspar's paper, where he says, okay, a protein, didn't evolve to have symmetry. A protein, the vast majority of proteins have no symmetry at all. So imagine that a protein is my left hand, no symmetry at all. There's 60 people here. So imagine I asked all 60 of you to come up and donate your left hand. So you put your left hand, okay, here's my left hand. You're shy, you're not coming up. I'm here, but imagine now you're 59 other hands around my hand, and I give you instructions. Take your thumb, and in this cloud of hands, find a pinky. Take your thumb and stick it on a pinky. For those who aren't born in the United States, pinky is the little finger. I don't know why it's called a pinky. It's called a pinky. And so you see here, thumb to pinky, and then over here, there's a thumb to pinky, over here, thumb to pinky. And that gives you fivefold symmetry. And then, and so you do that, you get a bunch of pentamers. And then I say middle finger to ring finger, okay? And so then here's a middle finger, there's a ring finger. Here's your middle finger, ring finger, middle finger, ring finger. You get a threefold symmetry, you get a triangle. And one final instruction, index finger to index finger. Since it's the same finger, it's twofold symmetry, okay? And then you, with those simple instructions, just local rules, you form an icosahedron. And so when we went to try to implement this out of origami, we said, okay, instead of starting with the 60 monomers and getting them to go together, that's going to be an engineering challenge. We're going to cut out a step, and we're going to instead synthesize trimers. We're going to synthesize triangles, a threefold symmetric object, and then that we're going to assemble. So it's already coarse-grained. We're not doing proteins, but so our fundamental building block is going to be a trimer. Actually, viruses often do this. They often form oligomers that then assemble, and I'll speak more about that later. And so now I had said that CCMV, calchloric mosaic virus, has 180. So what gives there? So now what you do, before your primitive triangle was contiguous lattice points. To form the CCMV, you go one step in the H direction, one step in the K, and then now you form a new triangle that's larger, and then you can cut out 20 of those triangles and then populate it with proteins and assemble. So like, look here at this inner vertex, you cut out along the black line and you glue together the edges. So along this black line here, you have 1, 2, 3, 4, and then 1 half red here and 1 half red there. So when you fold it up, you get 5 complete proteins. So that's a 5-fold vertex, that's the thumb to pinky thing. But in the center, you now have these, you see in the center of the triangle, you have these 6-fold. And they have actually 2 colors. And that's because if I look at one of these triangles, the 3 proteins, these 3 different colors, are in different environments. This one, red, has 5 neighbors. So it's in a 5-fold coordinated environment. These orange and, I don't know, more orange, more yellowy orange and more reddy orange, I don't know the names for these 2 oranges, both have 6 neighbors, but this is going to form a capsid. So there's an inside and an outside. So these units, even though they're in 2 dimensions, have chirality. So there's a directionality to it because there's an outside normal. So then you have, then this orange one is clockwise away from the red. If you go clockwise from the orange, you get to the red, while this guy, this redder orange, you have to go counterclockwise to get to the red. So each one of these proteins knows where they are and know they're in a unique environment. And so Caspar then realized that this T equals 3, triangulation number, is the number of distinct environments. So if you're going to engineer this, you need to give your proteins 3 different attributes. So it can be different bond strengths, different angles. I'll go more into detail to it, but there's 3 distinct environments that the particle has to respect. And he worked out the whole general framework. This tells you the triangulation number in terms of the lattice vectors, and then there's these quantized sizes. So let me pass around. Here's a T equals 3. So T equals 3 has 60 triangles. Whoops, sorry. And here's a T equals 1, icosahedra with 20. That guy can go around. And then this is one we made out of origami. This is a cryo-EM. This is not a computer animation. I mean, those are like toys. This is raw. This is data taken from cryo and reconstructed. And since it's data, you can look inside and see the internal electron density. So here's a cut through the capsid. And then we can do all the platonic solids. So here's a couple of octahedra made from the same thing. So, I mean, talks get boring. So here's some things to play with. And you can make, big domes. So this is Disney World. These are people. For sense of scale, these are people. Okay, we're interested in self-assembly. So what's self-assembly? You put a bunch of monomers of the virus in a cell, and then Brownian motion shakes it up, and then it assembles. So that's what this is. I bought on Amazon a transparent globe, and I 3D printed these guys and shake, and it self-assembles. And so here you go. I got them here. I didn't bring the globe because it makes an annoying rattling sound, and then you won't listen to me, you'll just hear the rattling. So I left the simulation, the CPU I left at home. But let me just assemble this by hand, and then I'll pass it around. And it's very easy to disassemble. It's a little bit harder to assemble, but you will rapidly get the hang of it. I think. Okay, so I tend to make 2 oligomers, the kind of the critical nuclei, we call these. And then I put them together to make the octahedra, and then pass that around. You can disassemble it. So each cylinder represents a double-stranded DNA. So this is an exact model of what we build out of origami. And so that was our inspiration. And now here's the, yeah, okay, you already collapsed it? Good. I told you it's easy to disassemble. But yeah, you don't have to assemble it. You can just pass around the pieces, whatever. I mean, don't feel stressed about it. I don't want you to be stressed over assembly. Just pass around the individual pieces. So what is DNA origami? Has anybody done DNA origami here? I mean, okay, people in my group have done DNA origami, but I mean, not from my group. Okay, so you, just very briefly, but of course, interrupt and ask questions. So you pull out of a E. coli, and we use filamentous phage, MFD, a 8064 single-stranded closed loop, circular loop of DNA. Single-stranded circular DNA, 8064 bases. You can buy it from New England Biolabs. This is a version, M13, MP18. And then so that comes from nature. You can't change it. Then you synthesize 200, what we call staples, oligos that are about 50 bases long, okay? And then the idea is that these staples, these oligos will take disparate parts of the circular loop and tie them together, pinch them together. And so you have 200 pinch points. And so you imagine gathering in this thread. You have this long loopy thing, 8064 circular loop, and now you gather it in with 200 pinch points. And, and then the first one by Paul Ruetherman, 2006, is a smiley face. And of course, if you make a smiley face on the nanoscale, this is these are whole, smile of tens of nanometers, you get the cover of Nature. Because while frivolous and clearly frivolous, it also tells you the power of this technology. There's no, symmetry here. Making a smile with dimples and eyes and things means that you have atomistic control. And this was the first time that you had that kind of control, that humans had this kind of control in, humankind over molecular nanoscale. And you could just design anything you want, frivolous things like this or. So this is 2006, the first origami. The question was, why are they flat? Because flat is not very interesting. We live in a 3D world. 2D world is pretty useless. If you can only make 2D objects, it's not very interesting. I wouldn't waste my time on it. But this was 2006. By the next slide, you could assemble into 3D. So here's an animation where the blue is a long circular strand, and then the little guys are the little oligos, and then you cool it down in a quench, and now you make a 3D object, okay? Because, you got a little cleverer, and there were a bunch of people who got super excited by the 2D thing, and in 2 or 3 years, there were a whole bunch of Nature and Science papers, and people were in 3D. And the folding, the secondary structure is holiday junction. So you have, and DNA folding is so easy compared to protein folding. It's millions of times easier, which is why I went into it in 2017, because I wanted to do protein design since I was a kid. In college, I took biochemistry class. After like 4 lectures, I dropped out. It's just 20 amino acids. It's and every 10 years, I would try to take a biochemistry class. I never could get through. And so in 2017, I said, okay, I'll work with DNA. The folding is completely understood. There's only one fold, and that's the holiday junction. And the holiday junction is, so imagine I have 2 completely separate double-stranded DNA. And now I just substitute the bases that are in this red loop. The bases here in this part of the red loop for the ones that are in this green loop. And then it just crosses over. And likewise, if I substitute these bases in the green loop for these in the red, it will cross over. And now I've tied together 2 strands. Biology uses this to make your chromosomes and stuff. And so this is called a holiday junction. That's the only rule. So you lay out your strands, and then whenever 2 helices face each other, you do a crossover. So the dark blue or violet is the, it runs off the page in each direction. It's the single circular loop that goes around. And then like this cyan is one of the staples. So you see it just crosses over and comes back. So you're just pinching together. And now this is from an all-atom simulation, which includes the charge of the phosphate on the backbone. And so there's electrostatic repulsion. So where they're pinched together, the Van der Waals forces and everything hold it together. And then when they're far away from a pinch, we call this chicken wire, they expand away due to the repulsion. So this is from a atomistic simulation. And then you can make, over the years chemical, you make all kinds of shapes, whatever you want, you can make. And so we made triangles. And here's a EM of our triangles. And here's a cryo EM of one of our triangles. And then you can do the all-atom simulation, and you can align it with the electron density of the structure. And if you find it didn't work the way you designed it, then you go back and you say, oh, it's this base didn't bind correctly. I'm going to engineer it and change it. So and then then you can think about, what about my binding sites and how am I going to assemble it? And then you have dihedral angles, slopes built into the side of the thing, and then you dock them together and analyze the binding angle, because you need to have control of the angle. And then the octahedra that I'm passing around, that this is the cryo-EM. So by adjusting the angle, dihedral angle of the side of the triangle, you can assemble different structures. And then we can also change the angle and make the icosahedral also passing around. And then you can see in the electron density as we're slicing through, you see the spiral pattern. Those are the Holiday junctions as the pairs switch, and we're on a square lattice, so you switch the helix goes every 90 degrees, then you cross over in one direction, then you cross over in the other direction. This is my shock and awe slide where we did cryo on a bunch of the different triangulation numbers from Caspar, including T9, but T9 was too big to even fit in the ice, and so we couldn't do cryo on T9, and then we have a whole series of papers on it. So why triangles? There's a mathematical theorem that any surface can be triangulated, and computer games use this, and finite element simulations use this mathematical fact. So that's why we said triangles, because triangles are generic. You can triangulate anything. There's a mathematical theorem that says you can triangulate anything.
So for those structures, how stable are they? Do they stay in solution or
So there's two answers to that. The question was, how stable are these assemblies? And there's two answers to that question. They're being assembled through weak forces that temperature and ionic strength control. So if you change the temperature or the ionic strength, raise the temperature, for example, they'll come apart. However, we can add moieties so that when we shine UV light on, they're covalently bonded. And it's actually, you don't have to add anything because the DNA is aromatics. And if you have two thymines opposing each other and you shine light, there's a cross section where they form a covalent bond. So you just take your thing, you put it in a UV for 5 minutes, and then it's permanent. So, and you'll see later in our studies of antivirals, that's what we did before we introduced it into the mice.
how generalizable are your dealing procedures? So if you have a given structure, you have to optimize it to use that.
I'm going to defer answering that until a few slides from now, where I will address that in some detail. So any manifold can be triangulated. Triangles are structurally rigid. So I pulled this off of YouTube a couple nights ago. So this cardboard thing he has is hinged. Each of the joints is a
How do we make
freely.
structures that aren't all
Oh,
wobbly?
his voice comes through.
Well, let's try. You might have tried this experiment at school. Almost any shape you choose is going to deform since there are rotational forces at the
So
joint.
that was 5. A pentagon is
Except
not
for a
stable.
triangle. The structure stays rigid, and we can
Completely
see that
rigid.
there aren't any rotational forces at the joints, because if there were, the joint would rotate, like in the other cases. Remember,
So
the hinge joints we're using offer no resistance to rotational forces whatsoever. That means the reason these joints aren't rotating is not because of resistance to rotational force, but because all of the forces are axial.
Yeah. So triangles are amazing. And so, squares will shear. You never frame a house out of squares. You always put a cross brace, otherwise your house will fall down. And that's why the Eiffel Tower is built out of triangles, because triangles are awesome. They're the only mechanically rigid shape. You make a pentagon, it's going to fall down. A hexagon, it's going to fall down. A square is going to fall down. Only the triangle. So that's why triangles. The other reason is that DNA and proteins like alpha helices have intrinsic chirality. And so if you, and there's this theorem from topology that relates the linking number, the number of twists in a wire, to so twist is how many times one thread, this is a double helix, twist is how many times one thread crosses over the other. And then writhe is how many times the thread of threads crosses over each other. So it's supercoiling. So if you people are too young to remember telephone cords, but when I grew up, telephones were attached by wires, if you can imagine, and they would always tangle up. So so I brought this for the younger generation. This is like my iPhone cord. So if I take the cord and I just put it together, it forms a nice loop. But if I introduce twist by rotating the wire, a number of times, and I'm not sure exactly how many, but I want to make sure it's dramatic. So I do a bunch, and now I put it together. You see, it forms. It writhes. So 1, 2, 3. There's 3 writhes, just like in there, and that. But the number of writhes and twists is a topological constant. And so, why does this matter for origami? The DNA has a twist in it. And so then if you link it up, and when you're doing these crossovers, you're introducing a torsional strain, and that can accumulate and cause the structure to rotate in unanticipated ways. So here's a simulation, all atom simulation, where I had just a thin triangle, and then I relax it, and it's just like a potato chip. It just flips over. While if I make it thicker and mechanically stable, then it resists the twist. So this topological mathematical thing relating linking number, twist, and writhe means that there's a propensity, if you don't compensate for the twist, to come out of plane. But you can overcome that, and that's very hard to calculate, and except trial and error, and then you have to test it in cryo. It's really a pain. Believe me, we did it, and it was a pain. And so then we realized, no, make it thicker, and just, it's cut the Gordian knot. Just, over-engineer it. It's not going to go out of the plane. So, and then a final reason of triangles is this mathematical theorem that there's a duality between dodecahedrons and icosahedrons. And what I mean by that is, if you Google, you find anything. So if you put points at the center of this dodecahedron and then you connect them to be a face, then you transform. So if I started with a dodecahedra on the left with these pentamer faces, and then I take a point at the center of each face, then I get a icosahedron. And if I start with an icosahedron and I put a point at the center, then I construct a dodecahedron. And so in the first work out of the Dietz lab, he started making vertices of origami to assemble into a dodecahedron. So he made these vertices, but then there was this uncontrolled twist, and the yield of assembly was just crap. And so I thought, okay, I can do this mathematical transformation to make an object with the same symmetry, but go icosahedral. And because it's closed, there's no possibility of adding a twist. So if I have, if my DNA is off by a quarter of a turn, the fact that it's one closed loop and comes back means that it can't get a net twist in it. It's topologically constrained. And so then it could still do the supercoiling, but if I make it thick enough, I'll prevent it. So I could get away from this design problem of having uncompensated. And we'll get back to proteins, where this manifests itself in protein design. Just a few words about Don Caspar, who passed away a few years ago. In the obituary in Nature, they talk about his theory of quasi-equivalence and how he learned from Buckminster Fuller, the story I told. A story I didn't tell was that he did his PhD thesis in the 50s on tobacco mosaic virus, one of these cylindrical proteins, and then viruses. And then he worked with Rosalind Franklin. And Rosalind Franklin is famous for taking this photograph. And so 2 days ago, I took this photograph and I put it into ChatGPT 4.5, and I said, please, this is an X-ray diffraction photograph. Please interpret it for me. And it just gave me the perfect explanation of what it explained that this, is the, anyway, it explained every feature in this. I didn't tell what the picture was. It says, this is photo 51 from Rosalind Franklin, one of the most famous pictures from structural biology. It told me in like 15 seconds. It's like a really good grad student, Anyway, so there was a play by Anna Ziegler about this photograph and Don about the story, because this photograph is famous because Watson and Crick got to see this photograph without the knowledge of Rosalind Franklin. One of her postdocs gave the data without telling her. And then once they saw it, they could deduce the structure of DNA. So they got the Nobel Prize, and she didn't. And so and then there's a play about this called Photograph 51. And when that play was shown first in the West End in London. Nicole Kidman played Rosalind Franklin, and Caspar was backstage. So that's pretty cool. And then in 1958, he created the first laboratory of structural biology at Children's Hospital, 100 meters from here. And his support for young scientists was extraordinary, as well as his advocacy for women in science. And in 1980, he hired me as a 22-year-old to do electronics to build the first generation of digital X-ray detectors. And so it was with pleasure that my colleague Mike Hagan and I showed Dawn in 2019 our first cryo-EM of the triangle, and then the icosahedral structure, all based on his architectural plans for viruses. So that is, I don't know, one of the highlights of my career, to be able to, like, a guy who started me and gave me my first job or second job, one of my early jobs, and launched my career, and then I could go back. So, okay, how do we commercialize this? So during COVID, my colleagues realized that you could take these shells, and passing around one of these half shells, and you could put an antibody on the inside, and then you could deactivate virus. So there are very few generic antivirals. And so you had to vaccinate the whole world. And then the whole world got really angry, and now is cutting NIH and the Center of Disease Control because we got them vaccinated. And instead, if you get bubonic plague, everybody who has bubonic plague, and they're getting those warts and boils and they're dying, is happy to take penicillin, which cures you in an hour. And everybody's happy to take penicillin, even though maybe they'll have an allergic reaction to penicillin. They're going to die from the bubonic plague. The chances of dying from penicillin are really, really small. And so it's always better to have a cure than a prevention. A cure is much better than a vaccine. So this is a cure for a viral attack. If your body's full of viruses, the vaccine, it's too late. But here we put antibodies on the inside of the half capsid, and then it's this kind of Star Wars type ship that then that gets the Death Star, and now the virus can't touch the cell. So let's say this is COVID. It's got 100 spike proteins. If one of those spike proteins touches your cell, it will fuse. It will release its genetic material. The antibodies have to hit all 100. They have to be 100% effective. Here, only one of the antibodies has to work. One antibody binding to the virus deactivates 100% of the virus, because it's the shell of the DNA around it that gives you the deactivation. So here's, we made a variety of these capsids, and this is with human, hepatitis B virus core being deactivated. And so my colleagues, Dietz and Siegel, then have spun off Captics in Munich to commercialize this concept. So I mentioned tobacco mosaic virus, the subject of Don Caspar's thesis, and that's a cylindrical virus. So you can play the same game that you do with icosahedral viruses. So this is a drawing taken from Caspar's paper. Back in the day, you did hand drawings, and it's really kind of cool. And so then here's your asymmetric protein with its binding sites. And now if you're going to make a cylinder, you just cut along the edge and you fold up. You just connect the blue dot to the blue dot. This again in his paper, where he actually literally did that and glued them up, took a photograph of it, and that's the figure in the paper. It's none of this Adobe Illustrator junk. And so, so then, okay, well, let's do that. So what are the binding rules? You need to have, you need to have the right geometry. They have to be the right angles, because you have to have negative curvature in one direction, positive in the other. And then we have single-stranded DNA as binders between the two to form it. And then basically, you just roll up this flat piece of paper and glue it together, and that's your cylinder. And so the thing is that any of these points on this triangular net will form a cylinder. It'll just be a cylinder with a different chirality or a different diameter, because there's only one species of triangles. And so just by changing the curvature in the sides of the triangles, you get these different diameters. But it's the same interaction matrix. The interaction is the red binds to red. And so you see two reds there, blue binds to blue, and white binds to white. And then it's only controlling the angle. So in this case, 20.8 degrees on this side, 28.8 degrees on this side, minus 10.5 on this side, that gives you the cylinder. And then if you want to make a cylinder of a different diameter, then you have to change the angle. But every possible angle, so when we assemble it, we've got a very polydispersed mess. That's because if there's any little fluctuations or any off in your angle, you still satisfy the specificity because there's only one species of triangles. And so, it's economical but imprecise. And if you have flexibility in your bond, then you're going to get lots of polymorphism, which is, if you're an engineer, that's bad. And so, that was using this economical, only one kind of triangle. But we see in biology, to get precision, you need to have many different species, this addressable assembly. So this is what we were doing. Let's say our target was to get 10, but we got 9, 10, and 11 because of flexibility. And so what you do then is you add specificity. So then you say, okay, only red binds to blue. And so it has to alternate. So now you have to have an even number. So you get rid of all the odd numbers, because an odd number has two reds, odd number has two reds. They're impossible to form. So by adding more specificity, by adding more, you get precision. But now it's less economical because now you need to make red species and blue species. So your cost to the company is doubled, but your precision goes up. And so, and now we didn't have this problem with the icosahedra because the symmetry of the structure requires you to have these T different numbers of environments. And so the icosahedra escaped that. And that's because it's a Gaussian curvature 2 closed object, while the cylinder has a net Gaussian curvature of 0, and every site is available. So kind of because of these deep topological reasons, the cure is the same. You add the specificity to the cylinders and you can cure the problem. So the problem is that if you just take a single species, you get a whole bunch of off-target states. And the number of off-target states goes like the circumference of the designed cylinder, C, squared over the elastic modulus. This has units of like energy. I mean, this is not energetic, this is not dimensionally correct, but that's the scaling. It's going to go like the area of the, the cross-section of the cylinder over the elastic constant. And so now if you have 3 species, so you make dimers, 2 reds, 2 blues, 2 greens, then you push out the allowed, like in the one was one-dimensional, one even odd, this is the 2-dimensional version. And then if you go out to 9 species, eventually you can have enough species so that the flexibility is small enough that you don't, by accident, get any of the off-target ones. And so we tested that experimentally by making, multiple species. And so if we make 1 component, we get this kind of polydispersity. Go to 4 components, it gets smaller, and then 16 components, it's 95% yield into your target. So by so you have economy versus complexity as a trade-off, and we have a precise mathematical formula for how these trade-offs scale. And then, so we say it's due to flexibility. How do we characterize that flexibility? So we do cryo-EM, and every one of these dimers that you see in the cryo has a different bending angle. And those bending angles are going to be, if it's an equilibrium, are going to be Gaussian distributed, where the width of the Gaussian tells you the bonding modulus. So we did a normal mode analysis, decomposed all the deviations, and so you get twist, bend, and stretch. and you can characterize all the elastic constants of the material. And so, I've been emphasizing symmetry up to now, and then there with flexibility, but symmetry is only one piece of the puzzle. There's also, for kinetics, it's what is the binding energy? If we have many, many, many bonds, do we make them all the same, or do we make some with different strengths? Is one better than the other? And so we did simulations where we had this triangle with 3 different sides and 2 bonds. Side 1 binds to side 2, side 3 binds to side 3. That makes it T equals 3. I think that's that big blue thing. Does somebody have the big blue? Yeah, no, not the blue one, the bigger one. That one, the green, it's green. That T3, that structure is made out of these triangles. And what we found is that there is 2, color is yield. So these are from molecular dynamic simulations. And so there's 2 points where there's high yield. And one of these points, this one corresponds to giving, having a tighter binding energy for the dimers. The E33 is the dimer bond. So you first form white to white, you get a dimer, and then you assemble 30 dimers, you get a high yield. And the other pathway is if you bias the pentamers. You make the side 1 bind to side 2. This is the thumb to pinky. So you get your 5-fold symmetry. And now you take 12 pentamers and you put it together. You also get a high. So this, while if you made them all equal, then there was a low yield. And so, this was a prediction. This was a simulation that hierarchical pathways improve assembly yield over egalitarian pathways. And so then, you do the experiment and theory experiment. And so you look in the pentamer pathway, you only see pentamer intermediates. In the dimer pathway, you only see even intermediates. And so when you do the egalitarian, there's just a zillion of intermediates. And so the thing is like, it knows where its minimum free energy is, this beautiful T3 structure, but there's so many intermediates in between that it just can't find its way. It's lost. It's really, but if you guide it by saying first form a dimer, then form, first form a pentamer, then give it a little guidance. It's like a graduate student. Try this, and don't try to do everything at once. Just try this one step, this next step. Then they get there. It takes 5 years, but they get there. Okay, so now let's move back to proteins. So here's, a Baker, right? You're not going to have a protein talk without Baker. So here's a paper, 26 February, 2025, so kind of recent. So he makes this monomer, and he says it's going to form this, awesome capsid. And then this is what they form. And of course, it's a Nature paper anyway, because, that's the way the world works. But, there's not much designability. And it's there, I would say there's several things wrong with this design. I mean, who am I to say there's anything wrong with a David Baker design? But were I to say that, I would say there's 2 things. One is that the edges, if you have any residual twist, it's because these are vertices. And the other is there could be flexibility. And they focus in this paper on the flexibility as the reason. And that leads to this polymorphism. In another Baker paper, again, they are using here 3 components. So they're moving up in the complexity stage. But again, like this one, this one's a closed form, but it's not a triangle, it's a pentamer. And then this one has open ends. And then in the same cryo picture, you see many different capsids all right next to each other. So there's no designability. You get, everything all at once. And I think it's back to these same kinds of rule. I mean, that's a hypothesis. Are there lessons for protein design? Can this thing we did in this really idiotic, simplistic, naive origami tell us anything useful about proteins? I mean, that's a hypothesis that I would like to test. Our suggestion is yes, these engineering principles are universal. That's a hypothesis. You have to, maybe it's right, maybe it's wrong. So what are the lessons? So origami basically lets you focus on bond energy, the direction, the valency, addressability. And so you can study self-assembly pathways, kinetics, and yield, and then you can get lessons. These are very hard to do in proteins, but from these studies, you can get some lessons that if you're bold or crazy, you can say apply to proteins. And so, we managed to design, these pretty awesome structures with these and we kind of validated it with molecular dynamics simulations. We found also that hierarchical assembly is robust and has high yield. We think that could be a principle that rather than egalitarian, where they're all the same, and we identified that it's avoidance of kinetic traps. We found that flexibility is critical for controlling polymorphism, and that by increasing the complexity through addressability, you can overcome that. And so those are the 4 lessons that I propose are transferable to the protein world. And as Shakespeare said, what's past is prologue. And so my colleague at Brandeis, Yang, who's here in the audience, is designing these elements kind of based on this, concept, and has expressed some of these. And when he gives his talk in this series later in the year, we'll see if this approach has any merit. Thank you very much.
Great. Seth, thanks very much for the really fun talk. I learned a lot. Maybe I'll kick off questions, maybe with something a bit philosophical. Just thinking about the complexity of DNA origami is kind of mind-boggling, but also not more mind-boggling than protein folding, perhaps. So just wondering, why do you think structures like this out of DNA didn't emerge commonly through natural selection?
Yeah, well, natural selection has its own mission, and the mission is to make more of itself. And so as long as you're fulfilling that mission, there's no need to do this other, to do origami. So I think, okay, to be a little more, I think, profound, is that the potential space of structures is combinatorially vast, right? If you have 20 amino acids and you have a chain of 200, and 20 to the 200 is, I don't know, bigger than there are any atoms in the universe, and biology is only sampled whatever, 10 to the 9 out of 10 to the 100 or 200. So 10 to the 10 out of 10, we've sampled nothing. Biology sampled nothing of the potential. And so, biology did a lot with nothing, right? I mean, the 1 part in 10 to the 100 is what biology sampled of the potential. And biology's doing a great job with that, but there's such a greater space. And so and that space is too big to navigate brute force, even with NVIDIA. But if you take some kind of universal principles, then maybe you can be guided in directions that are non-biologic, but the new tools of de novo protein design give you access to. And so you can look at things that just have not been seen living, and evolution would never get you there. And that's kind of our motivation.
Going from the complexity, from a complexity point of view, the talk was really cool. Now I have a lot of ideas. So this might be out of left field and have nothing to do with it. But have you done, or do you know anything that did like cellular autonoma or any complex system simulation where you have neighborhoods? Because this sounds exactly like trying to replicate a fractal where you just increase the thing and increase the size, and there's movement and discrete rules that you can create like an emergent property out of it. Do you know anything?
Yes, so there's a group, the Italian guy, what's his name? The Italian guy who does the SAT, oh God, I'm blanking on his name. There is a group that has developed an inverse design process where you say, this is a structure I want. What's like the minimum number of building blocks and their interaction network to get me there? And he has this thing, I think it's called SAT, Rosso, right? Russo, R-O-S-S-O. Is that, wish I was Googling. I think it's Rosso, Paul. And it's kind of combinatoric. It's a little bit brute force, but it uses some of these symmetry principles. And so there are people who have been worked on this. We call this the inverse design problem. It's, you know, it's like what chroma does or diffusion. You start with a structure, you want to find the sequence. It's that similar kind of problem. And so I think that group by Russo, I can send you a reference to it, but there is a systematic algorithmic way. We took a symmetry-based that's computationally very modest. I mean, compared to his approach, but his approach is general and approximate. Ours is like exact, but constrained. But there are algorithmic approaches, and the community, this kind of community of self-assembly, has a lot of focus on it. There's also some AI kind of versions too. So I can point you, and I can give you some references. Yeah.
Seth, I have a question while
Yes.
Chris is getting the mic. So your talk was, I enjoyed your talk. It sort of brought me back to when I was a grad student. And part of the lab that I was in was really interested in carbon nanotubes.
Yeah.
And so there's a lot of parallels
Yeah.
that you're talking about with carbon nanotubes. You have a lattice, you have your lattice parameters, and you roll up a sheet
It's
into
identical.
a tube. And for carbon nanotubes, depending on the chirality of the tube, you would get semi-conducting or conducting nanotubes. And I'm just curious, so like people like Jackie Barton at Caltech have said, okay, well, DNA is a conductor in a certain sense. Has anybody, so there's two questions that I have. One is, has anybody looked at different conductivity across these large DNA origami structures as a function of the handedness and the chirality of the DNA, the shapes? The second is, one way to actually purify carbon nanotubes is actually to wrap them in DNA, to actually purify certain chiralities of nanotubes, carbon nanotubes. Has anybody tried to incorporate carbon nanotubes into DNA origami to actually make sort of conductive nanostructures?
So there has been a whole kind of branch of engineering that's focused on the photonic and aspects and incorporated semiconductor, or gold nanoplasmon resonance and conducting things. Nothing's jumping out to my mind about the nanotube, but I don't focus on it, but I would not be surprised were there are things there. In the origami, it's not going to be that case because each of our triangles is this whole wound up thing. We don't have the contiguous structure, but one could design things like that if that was your intent. And so, I mean, that's the origami is a great platform. So if you had the idea, I want a whole bunch of DNA wires with this, whatever 6-fold symmetry running this way, then you could use like our kind of architecture to build something like that. But our thing is not connect. It doesn't have that kind of connectivity. Yeah, because it's much larger scale than the nanotube. So the mapping goes onto the nanotube, but then it's each triangle. So it's a meta level. Yeah, while in the carbon nanotube, each node in the lattice is an atom. In our case, each node is a triangle. So it's a rescaled.
Definitely bigger scale.
Yeah. But those concepts are universal. Yeah, it's exactly the same concept. Yeah.
Thanks.
What are the conditions to get them to assemble? Like, is it in solution?
Yes, it's all solution, water solution, and typically there's a little bit of magnesium because DNA likes magnesium, and then you can tune the temperature at which they'll assemble. That's up to you, and you need a certain amount of salt to keep it from blowing. Because the phosphate is just so charged, if you like go to low salt, it will like blow up, and so to keep it condensed, you need, I would not recommend going below 5 millimolar magnesium. I would not guarantee stability below that. So you need a certain amount of salt, and then, but you can cross-link them, as I mentioned earlier, and then you can put them in anything, then they're rocks.
And in terms of the cavity, is that non-polar, polar?
So, I mean, we make ours out of DNA, so it's all polar, but you can put whatever you want there. I mean, because you can have single-stranded stuff coming out. You could put cholesterol binding sites, so it functionalizes to lipids. I have a whole talk on that. You can, I don't want to say, but you can kind of do anything with this, that you want.
It's pretty cool.
Speaking of doing anything, so
Yeah.
can you engineer movement into the structure, like switches on and off, control release?
Okay. So that's difficult. That's a challenge. That's at the frontier. A few groups are working at it. There's been some limited, that's the frontier. It's like the same in proteins, can you do allostery? Can you do, and so it's at the frontier. There's a few examples, it's super slow. It's, limited, but there's some baby steps, I mean, that's the frontier. There are some possibilities, but that's really at the frontier research. Yeah, so it's possible, but that's really difficult. It's not this kind of thing. I want to say is like a technology where you're saying, we have robust, high yield, easy to do, economic. We were taking this kind of engineering approach to it to do the kind of things you're talking. That's like research science where you're gonna get it's more exploratory, but that's a very important question.
Two questions, kind of. First is, have you explored making these out of RNA instead of DNA in any sense? And judging from the description that you said of how they assemble, I can assume that at least making them assemble in a cell would be or producing these in a cell would be quite difficult. Is there any? Yeah.
So at Brandeis, and I think also in Munich, there's been no work on RNA, though I don't want to really speak for Munich because they've done everything. But there are groups, and particularly Arizona State University, the group of, I think, Solch, SULC, with a little Cyrillic thing, has done, that group has done stuff on RNA. There are groups working with RNA, and we have not, but yeah, and then there's more folds possible, and you can, and also some more enzymatic activities, yeah. But our group, Brandeis, has done none.
In a cell? That's the second question.
Oh, in the cell.
Yeah.
Yeah. So Hendrik has put them in mice, and they will clear out the virus over 24 hours, and they don't generate much immune response. And then they go out through, I don't know, whatever, kidney, liver, after 24 hours. But they're once you covalently bond them, you can put them in physiologic and they're fine. And they can't be digested either because they're all cross-linked and stuff. The enzymes can't get a hold on them. If it was just a double-stranded DNA, it'd get all chewed up. But when it's all wrapped up in these tight bundles, they're inaccessible on a time scale of a normal body clearing a drug.
You mentioned that holiday junctions are the primary motif used to stabilize these DNA origami.
Yes.
I was just wondering what you think, if there is a consensus, like stable local structure motif that would stabilize these de novo designed proteins. Seems like maybe helix packing is the approach you've gone.
Yeah, I mean, alpha helix are awesome, right? I mean,
yeah.
Yes. What can I say? Alpha helix are awesome.
I guess a question about this is a follow-up. I know when you were showing the holiday junction where you have these two double-stranded DNA, they swap. It kind of resembles, I think, what you see with these de novo designed helical bundles that might have domain swap dimers. Is this the approach you're taking, or is this more?
Kind of yes and kind of no. I mean, we're doing everything, right?
Yeah.
So that's one approach for protein assembly, but there's many other approaches, where with the interface engineering that you get from, any of the oligomeric proteins, right? So domain swap is one way to accomplish something like this. Other ways are engineering the interface.
Yeah. I guess
So,
the next
so we're
question-
doing, we don't know what's going to work, so we're trying everything.
And I was also curious of what dictates the size of these assemblies. Like you mentioned that, like I know protein's much smaller than DNA. That's why we have much smaller complexes. But is there a need to go smaller? And what goes into the design of making smaller complexes?
Well, I mean, for proteins, there's a need to go smaller because, you can only express something, I don't know, 400 or 500 amino acids, and then your bacteria goes on strike. So, there's those limits. But I don't know if that's what you're asking. Well, as far as I know, nobody pays attention to that in this community. It's like on or off. I mean, when I ask people, so, I've been to a bunch of these talks and I always ask them that. And they say, oh, we want to make picomolar KD, and then we can always screw it up and make it weaker if we want. So, we just try to make it as strong as possible. And then if you wanted less, I can just add, mess it up. That's the answer I get from everybody. Yeah. And so I think that's not quite the right approach, but, 'cause I'm biased from this work, but as far as I understand, that's a consensus of this community is make it as hard as you can. Let somebody else worry about making it weaker. Or maybe I mischaracterize it.
There's been other work with DNA, not just with like origami, but like computation. So like people have done things like play tic-tac-toe and other wild
Yes,
things.
yes.
Is it possible to combine those types of two technologies to kind of get DNA origami plus computation?
Yeah.
I guess in some sense, it's sort of like a previous person's ask about like hinges.
Yeah. So again, I think those are kind of engaging ideas, this kind of computational aspects, and certainly proteins have that also, right? I mean, switches, regulation, expression, there's, phosphorylation, there's many kind of switches, computation. So I think looking at things from that perspective, I think is fruitful, but I don't have a specific case study, but yeah, we think we kick in our, when flights of fancy, we talk about that kind of stuff, but yeah, I think that's the future, but I don't have anything now in the present.
Awesome. So before we break for more pizza and beer and just general chat, I have to ask maybe the most burning question of the evening. What was your first job?
Yeah, my first job was at MIT building a kind of a Coulter counter to measure antibody antigen interactions with a single nanopore where proteins were going to. This was like in the 70s. Yeah, the electronics was like really
awesome. Seth, thanks so much. And yes, please help yourself. More pizza, more beer, more conversation with Seth and each other. Yeah. Cheers.